have the review session for everyone.
I also reflect back on it at home, so I can re-listen.
That's a new one. Well, I've definitely recorded before many times, but not with a tripod. Shower. Okay, what do you want to talk about?
There's like a lot of stats in like throughout the, like in how many deaths and like percentages.
Oh, yeah.
Is that just for our information or should we memorize that? I don't know if you're gonna like, I don't wanna miss it if you're gonna ask like how many people died from like the flu or something. But like, I also don't wanna memorize it.
Yeah. Specific numbers are very unlikely to be on the test. They haven't been on any of the other tests. Yeah. But knowing ballpark, I think I would hope that people walk away from this class knowing kind of ballpark figures. Like, the flu affects a lot of people every year. And malaria and AIDS affect a lot of people. Other diseases that we hear about are also important, but for other reasons. Like, the classic example that I just went over on Friday was SARS virus. We heard a lot about that, and it still is maybe in the human population, but there are relatively few cases. Lyme disease, there's cases every year, but it's not like that's one of the most prevalent diseases, but it's there are cases every year, and it's interesting because it is passed from the insect vector, and it infects people, and when they're infected, it can be extremely debilitating. And it doesn't just infect immunocompromised people as well. So, I mean, there's, so knowing those kinds of relative importance and why we study them is important. 117,000 people died from this every year.
In my notes for taxonomy fungi, I have they are classically like separated by morphology and sexual sport progression.
Yeah.
But for modern day techniques, what do they just do gene sequencing, or how do they determine it now?
That's a good question. So now they do DNA sequencing for sure, but all of taxonomy has kind of, in reality, moved to polyphase, and it always has been polyphasic taxonomy, where people try to make these species designations and put everything in a hierarchy that is evolutionary based. But then there has to be some level of practicality as well. So the specific answer to that question ends up being different for different groups based on what the experts in those fields have determined historically or through an understanding of the organisms, what's the most important features to look at, what sorts of phenotypes are most important to look at in order to distinguish species or other taxonomic units that are important to distinguish. Like, in some cases we'll look at pigments, and in other cases that's totally useless. And in some cases we'll look at immunoreactivity of the surface proteins, and in other cases that may be a lot less important. So yes, lots and lots of gene sequencing. The big trees are always made with gene sequences. Then the kind of species descriptions and finer level tests are they often use different features of the microbes depending on what they are. And that was illustrated, I think, in the lab, right? Where you use some things for enterobacteria and other things for gram-positives like staph and strep. You're not, and that's just practical. We're not, for a while anyway, we're not going to be able to just sequence everything we come across. That may be in the near future. The sequencing is getting so cheap. But, you know, med techs and other lab people do those methods that you use in lab in order to identify species.
For this study guide question, plant pathogens threaten human health in two ways. Were there specific two you were looking for? Because we thought maybe with the toxins, and then if they infect crops and humans can't eat those foods. But are those the two that you had in mind? Or?
Those are the two I had.
Okay.
Were there any others? Any other candidate answers?
Not really. Just want to make sure that's...
Let me think about it. Those would be the two big ones. For human health.
Aspergillus is listed as being an airborne fungal pathogen, and it's also an intoxicant. But are the diseases it causes with those two methods like totally different?
Yes. Aspergillus is one of those organisms that's found everywhere. Like I think I mentioned that yesterday. So its primary habitat is probably as a decomposer that's kind of randomly found in the soil and on leaves and stuff. But it will also grow on animals. It's just super adaptable on a physiological level, but also, if you look into it, Aspergillus is actually a genus that, if I remember correctly, it is as old and sort of encompasses as much genetic diversity as mammals or something. I mean, it's just something that a lot of things have been dumped into, and they all share that name. So there's also just a lot of variation over time. All the different strains in Aspergillus. So they can grow in the lungs in immunocompromised people and cause sickness that way, and that's a big problem for especially immunocompromised people, AIDS patients, and others. And then they can also grow on plants. There's other strains that will be better at growing on plants, and they can produce those toxins. And they're also, they should be like primary or secondary saprotrophs. Primary saprotrophs. They're not super good at degrading lignin, but they are very good at degrading lots of other things. Yep.
For like in like bacterial autogrowth in 13/13, there's a lot of like weird charts to go into like green sulfur, non-sulfur bacteria and stuff like that. Should we know the intermediates in there?
In the electron flow diagrams,
Yeah.
you want to know if you should know all of the cytochromes and
Yeah.
quinones and
Well, yeah.
Those
should we,
things?
like, or is that just like what should we know?
you don't have to know all of those intermediates. You should know the other stuff about those diagrams, like what's the distinction between cyclic and non-cyclic electron flow, why they do those different things, what they're getting out of this process of this electron transport chain, and where the energy is coming from, things like that. But I'm not going to be like, in purple sulfur bacteria, cytochrome B contributes an electron to what atom on what molecule. That's not what this class is about.
YM shift, like going from yeast to mycelium, is it? I don't know. The impression I got was that it's if a fungus infects a human, it's normally like a mycelium outside of the human and then
Right.
grows as yeast. But Aspergillus, was that the only exception, that it could actually grow hyphae in the lungs?
It's the only exception that we talked about, yeah.
Okay.
I don't know if it's the only exception in the world, but most of the fungi that colonize us will grow as yeast inside of our bodies.
On the slide that has the reduction potential for particle sulfur and non-sulfur, the input for electrons is succinate,
Right.
so that would be organo, right?
Organotroph.
Okay.
So it's those organisms are actually photoauto-organotrophs or photohetero-organotrophs. So they can use organic carbon as an electron source and even sometimes as a carbon source. But they're getting energy from the sun. So
Yeah, my only hang up about that is that it says purple sulfur and non-sulfur, but on the previous slide, it says that purple sulfur bacteria are obligate photolithoautotrophs.
So the succinate in there would refer to just the other ones. It's an example. It's not universal.
So it could just be a different electron source
Yes.
that would be lithotropic for purple
Right, it would be sulfur.
or purple sulfur.
And it wouldn't have to be succinate necessarily for the organotrophy. It could be some other organic compound. They actually have thought to use environmental humic material that are around. Some of them will have connections to other sorts of long connections, micron long connections to other parts of the biofilm, which may have a different redox potential. They're doing networking. More questions?
Would it be possible for you to bring up, I think it was a third quiz about the immune system and go over like the different epitope questions? Like, I know one was like 5,000, one was like 1,000. Do you know what I'm talking about?
Yeah. Those were on one of the practice tests, right?
Yeah, I think so. For the
Two.
second one. Yeah.
Bring that up. Okay, do you have a copy?
Not with me.
So it's gonna take a while to bring up the.
I have one.
But it's like, how many
She has a cupcakes.
How many epitopes? We can do it this way. How many epitopes would a single B cell be stimulated by, or be able to respond to?
Isn't it one for each? Or-
One for each.
Yeah,
How many types of antibodies could a single B cell respond
five.
to? Produce?
Five, right?
5. And then the last question was how many antigens could a single B cell respond to?
Just one.
It would be just one.
Okay.
Okay, so then, well, let's take a population of B cells. Let's say we have 100 B cells. How many epitopes? And they're all naive, okay? How many epitopes could those 100 B cells respond to?
500?
No,
No,
100.
100.
100.
Okay.
Because it's, in this case, it's 1 per B cell, and we're just multiplying by 100.
Okay.
So we get up to 100. How many types, isotypes of antibodies?
That would be the 5, so it'd be 500.
That would be 5.
Just 5.
Okay, they can still all, each one produce one
One
or
for each.
those 5, IgA,
Okay.
IgG, IgM. If we combined isotopes, isotypes, and epitopes, then you could multiply the 100 times 5 and
Okay.
500. And then the last question was if you have a population of B cells, let's say 100 B cells, how many antigens can you predict that it would be able to, that population would be able to respond to?
As many as you have B cells?
That one's unknown.
That
There was
one's
an
unknown.
option on the quiz
Okay.
for you're not sure because it depends on the combination of epitopes among the antigens, right? And the
Mhm.
size of the antigens. Some have just a couple epitopes, some have hundreds.
Okay.
So it's really impossible to tell without more information. Okay, does that answer that?
Yeah,
All right.
thank you.
On the things to think about for the review, there's consequences of not storing a chromosome in the nucleus. Is that so transcription and translation take place at the same time, so you can have attenuation?
Mhm.
Okay. I wasn't sure of the consequences. I wasn't sure if it was like negative.
Negative consequences?
Yeah, like the positive consequences.
Yeah, positives are still consequences, yes. So, I guess another thing is the fact that they can be organized into operons. The genes can be organized into operons, right? And single mRNA molecules can be transcribed and code for multiple proteins. So they are under tightly coupled regulatory control since they're actually transcribed onto the same molecule. So what is it? What else is it about the bacterial genomes that allows that to happen? Both of those things: attenuation and operon structure. So yeah, they're not. It's not in a nucleus, but what else is different? It's circular. I don't think that is necessarily going to help with these things. Yeah.
There's only one. There's only one big chromosome as opposed to like having a, bunch of.
Well, yeah. What? Yeah, lack of introns. So that's really important for what the stuff we just talked about. The things that you guys said are true, but the fact that ribosome activity and RNA polymerase activity can be tightly coupled together has more to do with the fact that the mRNA molecule in bacteria is not processed at all before translation starts. So there's no introns that need to be spliced out, which is really different. And there's no like poly A tail that gets capped. So they can happen in the same place.
slides for malaria immunity, I think it said, there was a bullet point that said acquired immunity, and then in like children, where the, something about children. Let me look at it.
Something about children. I think the point is that it's possible for people to become resistant to malaria.
It said, can result in suppression of disease in high transmission areas.
Right.
With high childhood mortality.
Right. So it's basically the people that can't become resistant will often die out of the population earlier if there's a really high malaria load. So not everybody becomes resistant, but some people can, and then they actually don't get sick, or they don't get as sick as they initially did, as their body, their memory T cells and B cells against malaria build up.
So,
But if that doesn't happen, then they continue to be dramatically sick. And in areas with really high case rates, getting there, here we go, then sort of purge those people from the population in
so
childhood.
it's either you form acquired immunity or you die. Like, is that, what is,
Under those circumstances where the malaria is, you're very likely to be infected, multiple times as a kid. There's not very good medical treatment, so you're not going to recover because you get good medical care. Yeah, it's a pretty severe disease, and so a lot of children die from it.
Can resistance to malaria also be like genetic, like the kids who die off are never able to grow up and reproduce, so then the people who do have kids are the people who are more resistant to it, or is it not passed on like that?
Well, for the top bullet point here, yeah, that's known to be a genetically controlled trait, the sickle cell condition. For the second bullet point, it's not as well understood. So I don't really know. I would be shocked if there wasn't some genetic component to it, but it may also have a component that has to do with the particular mutations or genetic rearrangements that occur during the production of your B cells and T cells, that individual, because that's different between each person. So it may have to do with that. It may have to do with environmental effects. Doesn't seem to be. I mean, if there is a genetic component, it's not strong enough that those populations are just resistant to malaria, right?
I have a question from the sample questions, number 8. It says, which of the following require the virus to have an RNA-dependent RNA polymerase? And it says that all of the RNA viruses is the right answer, so it's not just the negative stranded ones that need?
No, the positive stranded ones need it too,
Okay.
right? Because
Why is that?
they need to produce negative stranded RNA in order to make copies of their genome. They can't, right? They need lots. If they're going to produce a bunch of new virus particles, they need a lot of copies of their positive stranded RNA genomes. And in order to make those copies, they have to be transcribed, or not transcribed. They have to be copied from the negative stranded RNA.
So any RNA virus needs that polymerase?
Except for the retroviruses.
Okay. Okay.
So is that the RNA dependent DNA polymerase?
No, that's the retroviruses have. And so that's another option. So retroviruses can reverse transcribe to DNA. If it's not a retrovirus, though, and it is an RNA virus, then it has one of these RNA-dependent RNA polymerases. Okay.
So for like a retrovirus, when it comes in the cell, it's transcribed to DNA, or I guess it's made to DNA. And it's gonna make back to RNA again when it leaves the cell in
Yes.
the new virus.
Right.
Okay. So whenever it comes in, it's always what leaves in the new virus.
Yeah.
On that slide, didn't it have RNA-dependent RNA polymerase and then RNA-dependent DNA polymerase? What's the difference between those two?
The difference is, in both cases, you're starting with RNA, and in the RNA-dependent RNA polymerase, another RNA molecule is being produced. In the RNA-dependent DNA polymerases, it's producing the DNA.
Okay.
That's the difference.
So RNA-dependent DNA, it's the same thing as reverse transcriptase.
It is synonymous with reverse transcriptase.
Does it matter if you have positive or negative RNA to start with these dependent ones? Or can we just start like, you have to have negative RNA to use RNA-dependent DNA polymerase?
I don't think so. I mean, they end up making a double-stranded DNA molecule anyway.
Okay.
I think most of the retroviruses are positive, like it's shown here. But there's nothing, to my knowledge, biological or biochemical that would require it to be positive, since it's going to make both strands of DNA anyway. And then, I mean, I guess it's a shortcut, because then all the RNA molecules that get transcribed from the DNA can both serve as a template for protein production and can also be packaged in the virion. So yeah, I guess it is more efficient for it to be positive.
So for retroviruses, it doesn't matter if it's positive or negative, because HIV is listed as negative here.
That's a mistake in my slides. It's shown here in this figure as positive. I think it's positive.
But ultimately, it's like, does it matter then if it's positive or negative as a retrovirus?
Well, it only matters in terms of the efficiency of the process. So we just talked about that, right? At the end there, you've got positive RNA, and the positiveness means that it can be used in translation, right, to produce the protein. So if it starts off positive, and that's what's in the genome, or sorry, in the virion, then the positive stranded RNA that gets transcribed from DNA can serve both purposes. It can be put inside the virions, and it can also be used as the template for proteins. So it seems more efficient, right? And so if it was negative stranded and they wanted negative stranded RNA inside that virion, then that would require another step here. It would either require transcription in the other direction to produce the negative stranded RNA, or it would require one of the special RNA-dependent RNA polymerases found in those other RNA viruses to take this and produce the negative strand.
So positive RNA can be used for proteins, whereas negative can only be used to make another positive?
Yeah, right. Negative just means it's the non-coding sequence. Other direction.
Does the virus usually carry the polymerase, or does it hijack the cell's polymerase?
It will hijack the cell's DNA polymerase. interfaces, these have to be coded for by the virus. This has to, these bottom two have to be viral genes. So it's the DNA viruses that can use the host DNA. Okay, Alex?
When, so, what, going off of what you just said, they need the RNA-dependent RNA or DNA polymerases to be, or is that in the virion, or is that made inside the infected cell from the DNA, or from the genetic information of the virus? Like, in other words, is it
Yeah.
passed with the virion, or does it get made after the host cell has been infected?
I think it's often packaged with the virion, inside the virion. But not always. And there's a case here where it doesn't necessarily have to be. I guess a couple of cases. So if what's packaged is positive RNA, if that RNA molecule is really good at fooling the cell that's being infected, then that could directly serve, like is being shown here, to produce proteins, right? And if that first protein is an RNA-dependent RNA polymerase, then the protein can turn around and make this negative stranded RNA molecule and more positive stranded RNA molecules. So that's dependent on this one molecule coding for the RNA-dependent RNA polymerase and being translated as soon as it's injected into the cell. And so flu is not like that. For some reason I thought that it was. We had an example like that. Maybe not. You know, neither of the ones that we talked about in great detail, HIV and flu, are like that. I think it's possible, but not as common as just having the enzyme in there, because then it's best adapted. Taking more copies immediately.
It's usually in the virion.
Yes. And it is for those two, in particular flu and HIV.
Did we talk about hepatitis virus, A or B?
I had to skip it. I was running out of time. It still ran. I think I skipped it. We didn't talk about it, did we?
It's on the tag syllabus.
It is?
Yeah.
I don't think we'll have any questions. I think we've covered it. But West Nile is on there. And RSV and SARS.
There was one other thing, I think the very first thing on the taxa list, I don't remember going over, it was called like, it was a bacterium called like Verruca bacteria, and I mean, I looked it up, it said it was, like recently discovered and it could, I think,
Yeah.
oxidize, but I don't know if that we really went over it in
I
class.
think that Verruca microbium is a phylum
Yeah.
that, like you just said, has been recently discovered, and there is a slide that I have where I list a couple of those phylum and talk about how ubiquitous they are and how much there is left for us to learn about microbiology. And yeah, we don't talk about Verruca microbium in any more detail than that. It's amazing that it's found all over the world in almost every soil sample that you look in, and lake sample. We didn't know that it existed until 15 years ago. The same for acidobacteria, and the same for planktomycetes. And then I have a slide which shows a little bit more detail about planktomycetes and its importance. Yes?
Number 4 on the sample questions, it asks what is an adaptation that a fungus may have to deal with or reduce by levels of resources. I couldn't really find that anywhere in the notes.
Is that number 4 in the essays or the multiple choice? Essays. What is an adaptation a fungus may have to deal with reduced levels of resources? Describe the property of a fungus that would allow one to become one of the largest single organisms on Earth. You guys hear that story? No? Huh?
Isn't it like on a tree and then like spread for acres
Yeah.
underneath the soil?
Yeah. There's a fungus that in the soil covers like 100 acres or something like that. I mean, so I think it's bigger than that, actually. Hundreds of acres. It's up in the Upper Peninsula in Michigan, and it sort of is a pathogen, and it's spreading from one tree to the next, affecting the roots as it goes. So that is related to this adaptation that a fungus may have to deal with reduced levels of resources. So, does anybody know the answer to that? What are some adaptations that the fungi have to reduce levels of resources? Yeah. Spores? Is that what you said?
Yeah.
Yep. So that's part of it. That's one thing, right? That allows them to spread out, become dormant, and wait for a better time, or spread to other environments passively, right? Wind or something. But that's not what's actually helping them become one of these largest organisms, the largest organism on Earth. Because then you've got multiple, we don't refer to them all as the same organism. We have got multiple clones. They're separate. Yeah.
Yeah, like alternate metabolic pathways. You can just like go down the checklist, like one doesn't work.
Fungi, I mean, there's other organisms that probably do that better. But they can express different enzymes and proteins depending on the, and grow on different substrates depending on the environment. They're all chemoorganoheterotrophic. Some can switch a little bit to pathogenic or actomycorrhizal or saprotrophic. So yeah, they're flexible in terms of that. But what's the other? So what else? What allows a fungus to be the size of this room? And no matter how much exercise I do and how much I eat, I will never get to be the size of this room.
The mycelium.
Yeah. It grows as a network. It can grow, has this sort of indefinite growth form, and all of those cells are networked together. They can pass. So the adaptation is that they can pass nutrients from one part of that mycelium to another part. And end up what we call integrating over the environmental heterogeneity. So you've got environmental differences between different parts of this room, let's say. And in one area, fungus may be limited by phosphorus, and in another area by nitrogen, and a third area by carbon. And it can integrate all of those and grow better because it is able to explore and take advantage of different spots. So we do that by walking around, right? Animals are mobile. But fungi can't do that. So they instead have this network. That's what I was getting at there. Yes.
I had a question about number 7 on the test, and it said, how does the source of new genetic variants of the influenza virus and HIV differ?
Mm-hmm.
And I know on the slide it talked about how the influenza virus becomes like genetic drift or genetic shift,
Right.
but I didn't know about HIV.
Anybody else? No? Yeah.
Isn't it like influenza has different molecules in its genome, so it's more likely to recombine and do a
Right.
genetic shift? That HIV isn't really going to do the shift, it's just going to do the drift.
Right. And its drift happens fast. It's a reverse, it's a retrovirus. It uses reverse transcriptase, which is, even though the RNA viruses, the RNA polymerases don't have proofreading activity. I think their reverse transcriptases are even worse. And you get mutations that will accumulate within an individual person. So the drift happens, the mutation rate is really high.
It's high in HIV.
In Asia. It's high in influenza too, but it's higher in Asia. The other place, the other thing you could argue is that some of the genetic variation in flu will also be coming from other whole populations. So we have this constant barrage of genetic variation from flu that's circulating in birds and swine.